Our next speaker is Professor Mike Briggs, and Mike started off his career researching osteogenesis imperfecta, also known as brittle bone disease, and then went on for a variety of positions, including Wellcome Trust Senior Research Fellowship. And in 2012, Mike was appointed Professor of Skeletal Genetics in the Institute of Genetic Medicine at Newcastle University. And most notably, Mike is coordinating a huge project called Sybil, which is an FP7 funded project involving 18 partners over 5 years. So Mike is going to talk to us about repositioning of small molecules for inherited connective tissue diseases. Over to you, Mike.
Okay, thanks. So I'd like to thank Nick and Flora for inviting me here today. So I really wear two hats. In one respect, I'm a human geneticist, but really a lot of my training is in biochemistry, in particular cartilage biochemistry. So our research has really, over the last 20 years, has focused on a group of diseases due to defects in connective tissues. So I just thought I'd start by giving you an outline of my talk. I mean, obviously, these are a group of rare diseases, and I'm not sure your familiarity with them, in particular the skeletal dysplasia. So I'll spend a little bit of time discussing, introducing these diseases and putting them into some kind of historical context. But also, one of the approaches that we've been taking is we heard just recently from Bruce that obviously these 8,000 diseases, getting therapy for 8,000 diseases is very difficult. So we started to look at common disease mechanisms. Can we look at diseases genotype independent and begin to group them in the basis of similar diseases and could target a disease mechanism? So I'll talk a little bit about that. And our main disease mechanism that we've been focusing on over the last few years is endoplasmic reticular stress. And can that really be identified as a very good target with good therapeutic potential? Now, I'll give one or two examples about some of the recent work we've been doing on repurposing existing drugs. And I have to say, I admit now that most of the talk will only be talking about the approach we're taking, because we've not got very good examples yet of repurposing drugs in human connective tissue diseases. And I'd also like to mention a little bit about biomarkers, really, because I think they're vital and they underpin any future clinical trials, particularly if you're looking at long bone growth over a period of years. So really, inheritable connective tissue disorders have been recognised as a group of diseases for at least 50 or 60 years now, and we think that there must be over 500 of these that really affect a broad group of tissues, so the cartilage, skin, bone, muscle, tendon and ligament. Really, I think it was Victor McCusack, probably back in the early 60s, really started to identify these as a group of diseases and began to really group them together as potentially very similar. And in his first really book, this is the third edition from 1966, we can see that here are the diseases that he lists. AKU is obviously listed, and amongst them, right at the bottom, in other genetic disorders of connective tissues, is the disease that we're interested in, which are the genetic disorders of the osseous skeleton. So that was over 60 years ago. More recently, we can see that this whole group of diseases expanded. So you can already see the increasing complexity of studying human connective tissue diseases. Again, many of these you'll recognise. We've got the Ehlers-Danlos syndromes, there's probably about 8 different kinds, even more than that. OI, I think we're up to at least 10 or 11 different forms of osteogenesis imperfecta. Same with epidermolysis bullosa, quite a few different forms. But at least now skeletal dysplasias are beginning to occur as a single chapter within this book from 2004. So really, what are the skeletal dysplasias? They're a group of diseases, probably about 450 different phenotypes, and there's extensive clinical and genetic heterogeneity. They're defined really by short-limb dwarfism, but many of these conditions also, the patients also suffer from early onset osteoarthritis, which often requires hip replacement by the time they're in the mid-teens or certainly the early 20s. Although many of these are individually rare, as a group of conditions, you probably have an overall incidence of about 1 in 4,000. So it's not an inconsiderate number of different phenotypes. The disease is still difficult to diagnose at the moment, particularly for the non-expert, and there are currently no treatments for any of these diseases. And this really does result in a significant pain and disability, obviously leading to poor quality of life and really high healthcare costs. So this just really gives you the list of the range of different phenotypes that we're working with. They've been grouped into 37 different phenotypes. different phenotypic groups, and these are really based on clinical presentation. In some cases, particularly with the type 2 collagenopathies, diseases resulting from mutations in the same gene are grouped together. So I've just put this up really just to show the complexity of this range of diseases that we're trying to investigate and come up with potential therapies for. This is a meeting of I should say that the Little People associations in the UK are very well organised. The patient support groups are really very good and very organised. There's about a dozen in the UK. And this is a meeting that was organised in September by several of my clinical colleagues up in Newcastle. And it really brought together a whole range of people with these conditions to try and discuss the medical complications and really what they wanted out of research for themselves. And I'll come back to this because it's actually, for me, it was a very profound 2 days, and it's really, in many ways, changed my way of thinking and how we need to approach looking for therapeutic targets, particularly for rare skeletal diseases. So this slide really, again, gives you the complexity, the level of complexity that we're having to deal with trying to understand disease mechanism. So we now know that there's probably just over 450 different diseases. As I said, these have been placed into 40 different groupings. About 300 conditions, we've now identified mutations in 226 different genes. So again, a vast repertoire of different genes, probably all resulting in different mechanisms. And of course, that level of complexity has again increased because we have mutations in a gene that can either be dominant or recessive. So some diseases can be dominant, some can be recessive. We've also got a whole range of different disease mechanisms that we've got to consider. Non-alleles, haploinsufficiency, right through to really severe antimorphic mutations, so dominant negative mutations. So these are missense mutations that affect how a protein folds, and that protein may function in a complex with other proteins. So you've got a vast array of different disrupted interactions. What we see with these diseases is both allelic series and genetic heterogeneity. So by allelic series, I mean that mutations in a single gene can cause a range of different diseases. So mutations in collagen 2 can cause anything from a lethal phenotype through to a very mild chondrosplasia, predominantly presenting with osteoarthritis. We've also got genetic heterogeneity. By this, I mean that mutations in different genes can cause the same phenotype. So that again may have implications for genetic counselling, diagnosis, for coming up with specific genotype dependent therapies. And finally, we know that there's a lot of phenotypic variability, particularly intra and intrafamilial variability. So there's clearly genetic modifiers that are affecting phenotypic severity, and that's something that we've also got to consider when we think about therapeutic targets. So it's a big group of diseases with complex disease mechanisms. And I think that's probably reflected in the number of clinical trials that are currently under place for growth disorder. So I took this off the web. It's a 2003 report from just showing orphan drugs in the pipeline. So phase 1, phase 2, phase 3, and submitted applications. So here we have genetic disorders, which I guess are doing quite well at 85. But growth disorders really probably doing very badly. It's only 7 potential drugs under clinical trials. And really the only one that is actually for a rare genetic disease is the Biomarkers C-type neuropeptide that's currently under phase 1, possibly phase 2 trials at the moment. And I think that this really reflects the difficulty that we've got in trying to identify disease mechanisms and potential therapeutic targets. So coming back to what Bruce said earlier about these 8,000 different diseases, and obviously we're in the era of the genome, and everyone must have heard of the 100,000 genome project, and the big push on the government to sequence these 100,000 genomes from cancers and rare diseases, and obviously we're quite involved in that in Newcastle. But really, as Bruce said, you can't come up with 8,000 different diseases. I think coming from a genotype point of view, it's going to be very difficult. So what we started to do over the last 5 or 6 years really is try to identify diseases that may have similar common mechanisms and really try to target that mechanism. And it's possible that a drug, we could repurpose a drug, that may be effective for a range of different diseases that you would never have connected clinically. So it's the disease mechanism that we're really beginning to focus on. So we started this maybe about 7 years ago, and as Nick said, we've been quite fortunate in our research field in that we've probably had about 20 million euros worth of funding from the EU over the last 10 years, really start to work within Europe to try and understand disease mechanisms and come up with good diagnostic protocols. So this is a project that ran for 3 years, and there's several partners, and we really looked at 10 different mouse models. What we wanted to know is, were there any common disease mechanisms shared between these disease mouse models? So there's two things first. Being able to study skeletal dysplasias in a mouse model is absolutely imperative. There's no way that you can study long bone growth in a tissue culture model in the lab. So it's imperative that we have good, physiologically relevant mouse models. And the second is that we identified disease mechanisms, and particularly disease mechanisms in common, that we were not expecting to see. So some of the ones, some of the ones that you might see is changes in proliferation. That's something that we didn't expect to see, and we saw that that was shared between a number of different diseases. We also saw that ER stress was also shared between a number of different diseases, as was sulfation of proteoglycans, disrupted Indian hedgehog signal and so forth. So that really gave us, this small 3-year project gave us a good idea that we were on the right track and we could identify common disease mechanisms. We've since scaled this up, and we're fortunate to get one of the last FP7 funded projects. So this started just over a year ago. Again, we're coordinating it from Newcastle. We've got 18 partners with a budget just over 12 million. And again, what we want to do is we really want to take a very broad group of both rare and common skeletal diseases and try to identify common mechanisms that we can begin to target for potential therapies, including repurposing of molecules. So really, our objects of this project, CIBL, is to develop and validate a portfolio of disease models. Because without disease models, you can't do any repurposing of drugs. So we're going to develop over 200 new cellular models of skeletal diseases, 25 novel zebrafish, and also about 20 mouse models, and we're going to supplement that with about 25, 30 mouse models that we've already generated and validated. We're going to do large-scale genotyping and phenotyping, and we're going to use standard technology so that any mouse, so a partner in France, a partner in Newcastle, a partner in Germany, we're going to phenotype these models using the same way, using ontology, so that we've got the disease mechanisms and the phenotype really precisely described. Then we're going to use large-scale omics profiling. So we've got an omics knowledge factory. We want to do transcriptomics, proteomics, metabolomics, and really start to identify common mechanisms. We're going to underpin this using systems biology. So we're going to take disparate phenotypes and try and identify common mechanisms. So systems biology is really going to be the technology that will underpin all of this. So hopefully 3 or 4 years from now, we'll begin to identify how we can group skeletal dysplasias together and target particular mechanisms. So what I want to do now for the second part of the talk is really talk about the mechanism that we are particularly interested in Newcastle, and this is endoplasmic reticulum stress. So obviously the endoplasmic reticulum is the organ in the cell that translates all of the protein and folds the proteins for export. The problem is you can get ER stress. And ER stress can occur due to viral infection, to chronic inflammation, such as in asthma, hypoxia, sort of like ischemia, but also, particularly relevant to rare genetic diseases, is the accumulation of misfolded mutant proteins. And any of these four triggers can induce something called the ER stress response. Now, the ER stress response is now we're now aware that it's associated with quite a few number of different diseases, particularly neurodegenerative, cerebrovascular, in our case musculoskeletal, but also cardiac traits. So it's been identified as a common mechanism across a broad range of different diseases. So in this respect, these different diseases are all unified by ER stress. So this makes ER stress a very attractive target for new therapies, new therapeutic targets, particularly the repositioning of existing drugs. So I say this is really the tissue that we work on. So here's a radiograph of a bone. We've got the metaphyses, the epiphyses, and this region here that you can't see on the radiograph is the cartilage growth plate. It's non-mineralized tissue. So here we see this is just a growth plate stained with an antibody to a cartilage protein, and each blue ones, this is a cell nuclei. You can see that these cells are very neatly aligned. And if these cells aren't aligned, and if you don't proliferate correctly, you don't get long bone growth. So this is really our tissue of interest, and this is the, the organ that we really study looking at disease mechanisms. And I've just included a couple of pictures here, and I promise that this is really the only data I'll show, just to give you an indication of what ER stress actually looks like. So here we've got 4 chondrocytes in a wild-type mouse growth plate. You see the cell here. Here's the nucleus. It's not very clear, but you'd have these nice little ribbons of endoplasmic reticulum and some Golgi here. So we've got these 4 cells neatly aligned. Here we've got, here's the rough endoplasmic reticulum here. This is growth, these are chondrocytes from a mouse mutant growth plate. And you can see immediately that morphologically they're very different. So the cells are different shape and they can't align correctly. The reason that they're different shape is that you see this large area here. And this is because the endoplasmic reticulum is enlarged with mutant misfolded protein. So this protein, instead of being in here, it should be out here, making the matrix. But it's retained. And it's retained because it's misfolded and it's accumulated. This is just a colour rendition of the same images. Here we've got the nucleus in blue. You see the nucleus in blue. Here's the rough endoplasmic reticulum in green. And you can see immediately in the mutant, it's just full of rough ER. And that's going to have a severe consequence, profound consequence on cell phenotype. And what we see triggered is a process known as the unfolded protein response. And this is an evolutionarily conserved process. It's a signalling cascade that's meant to protect a cell against ER stress. It's evolved to deal with short-term stress, so heat shock, viral infection, hypoxia. But it's not evolved to deal with is rare events. And those rare events are rare mutations that cause a mutation to misfold. What we see is that in short-term stress, that this process here, this pathway here, I'm not going to go through it, is cell-protective. So it allows the cell to overcome this short-term chronic ER stress. The problem with rare mutations is that this stress is continued, it's prolonged for many days, for many years. And in fact, this process, this pathway turns from being cell-protective to very detrimental, and it causes cell death. So the accumulation of protein eventually causes cell death. ER stress has now been recognised as a mechanism for many different diseases. This is just a few, obviously cardiovascular. This is some of our work on chondrodysplasias. ER stress has been recognised as a disease mechanism in certain kidney diseases. So the misfolding of uromodulin, which is the most abundant protein in urine, causes ER stress and kidney disease. Certain genetic forms of stroke, again, result from mutations that cause misfolding of a particular protein. Myopathies, for example, collagen 6 myopathies, Bethlem and Ulrich, again, result from misfolding of mutant proteins that cause ER stress. Epidermolysis bullosa, and even some forms of deafness and also blindness can result from ER stress. So it's a disease mechanism that's across a broad range of tissues that affect a broad category of diseases. And this is really the mechanism that we're targeting in genetic skeletal dysplasia, but also in connective tissue diseases. So where are we at the moment? Well, we've got some very good model systems. So I said we've got culture, we've got cell culture models, but more importantly, we've got the preclinical mouse models that we've generated and validated. And I think without both of these, it'd be very difficult to ever really try to understand whether we've got good therapeutic targets that can be tested. We know an awful lot about the disease mechanisms, so we can actually study the effects of these mutations at both the molecular level, so we can see how these proteins aggregate and misfold, and therefore accumulated in the ER. We've got cell culture models that allow us to study the effect of these molecular events on cell viability and cell proliferation. And finally, of course, we've got the mouse models, so we can actually really study in detail using assays that we couldn't do in cell culture models. So for us, cell proliferation is probably the most important mechanism for long bone growth. We can only do that in a mouse model. So we'll be able to look at all of these different aspects. As well, we think we've got some really good, robust readouts. So we can measure the misfolding, the secretion of the proteins, but also the cell proliferation. So I think we're in a position where we can really start to, having got the model, we know the disease mechanism we're going for, we can start looking at potential small molecules of drugs for repositioning. And unfortunately, that's where we kind of lose a little bit of momentum because of the nature of the diseases that we're working with. So I'm going to start off actually by giving a positive story really on repositioning. And this is work that's done by a good friend and colleague of mine, Tom van Agmel in Glasgow. So he's interested in basement membranes. So he's a basement membrane biochemist. He's really particularly interested in collagen 4 and how mutations in collagen 4 cause these rare diseases. They're actually very reminiscent of these hemorrhagic strokes. So he's got both cell models and mouse models. In this mouse model here, this cell model here we've got the patient, and these are very similar images. So I should actually say, I'm going to take a step back and say that, so Tom works on basement membranes and collagen 4. We work on cartilage and cartilage-like proteins. But actually, we use the same techniques, the same experiments, and we share reagents. So that really shows you how common the different diseases, but the mechanisms are very, very common. Here's an image that I showed earlier, the accumulation of a misfolded protein here. And it's just labelled with PDI, so it shows in the rough endoplasmic reticulum. Treat this cell line with this particular drug, this type of sodium phenyl butyrate, and that protein accumulation is reduced. Okay, so if we're saying that protein accumulation is a disease trigger, by reducing it, that's going to have a good positive effect. And indeed, that's what you see. These are all markers of ER stress. So we've got BIP, we've got calnexin, we've got ATF4. And the expression of all of these markers of ER stress is reduced on treatment of this cell model with sodium phenyl butyrate. So that's one good example. Without giving anything away, I should say that Tom's now studying this in a mouse model, and I'm sure he'll be publishing it pretty soon. On the other hand, we went straight, probably not a very good experiment, but we went straight into the mouse model. So we've got a really good mouse model. We were studying it at the time. We thought this would be easy. We'll just introduce this drug, sodium phenyl butyrate, into the mouse chow and see an effect. We saw a marginal effect. So we saw the reduction in markers of ER stress, and we know that we've reduced ER stress. It's debatable whether we reduce the accumulation of the mutant protein, and we certainly had no effect on long bone growth. So for us, it was quite a disappointing experiment. One of the problems that we've got is that cartilage is an avascular tissue. So there's no blood vessels. Anything that gets in or gets out has to get in and out by diffusion. Sodium phenyl butyrate is quite salty. So if you increase the concentration too high, the mouse won't eat the food. So that's one problem that we've got working with cartilage, is actually you can identify perhaps molecules, you can identify targets, but getting it into the cells that matter, the delivery method, again, is going to be very difficult. We do know that certain small molecules can diffuse into the growth plate because we inject mice with BRDU, which is a thymidine analogue, and that does diffuse into the growth plate. So that's one experiment that we did, and obviously it wasn't very good for us. So we took it a step back and started to look at, this is just sodium phenyl butyrate, so it's obviously been used for decades. The urea cycle disorders have a very great safety record. I think people are beginning to think about it for cystic fibrosis, cancers and dementia, and I think it has FDA drug status for spinal muscular atrophy. So it's obviously a very attractive candidate to try and repurpose for some of these diseases. But the approach that we took was really to take a step back and start to use cell model systems again, and start to look at a range of different compounds. Now the way that we do this, we do a literature search. So it's not very comprehensive. We identified a dozen potential molecules that we could try, and we just test them in cell culture model. And what we found is, again, another level of complexity, in that if we're targeting the disease mechanism, it should be genotype independent. Unfortunately, these two different diseases, I've just labelled type 1 and type 2 for simplicity, different genes complete the same mechanism. Unfortunately, the drugs have different effects. So for this one here, no matter what small molecule we tried, it really had no effect whatsoever. In this one, we did actually have some success with this small molecule, and again, we've gone into the mouse model, and we're very excited about that, but it's a bit premature to say anything about that. So really, again, this highlights the problem in that we're back down to genotype specific. We've identified a common mechanism, but it's again genotype specific. So I think we're going to have to go back to the drawing board a little bit and even stratify out, if we've identified ER stress and misfolding of a protein as a mechanism, the way that mutant proteins misfold is again different. So I think we need to do a little bit more basic science on that. So I'm just going to put this example up because this is a recent study that was just published in Nature from a Shiro's group in Japan. And I think this was quite unexpected. It's incredibly exciting. This group had a range of human iPS cells expressing mutations of FGFR3, which cause either achondroplasia or anatophoric dysplasia. They treated it with statin, because statin is a known anabolic inducer of chondrocytes, and lo and behold, it had a positive effect on the cell culture model, and when they fed it to the mice, here's an achondroplasia mouse, it increased bone length. So they're not sure of the mechanism, but obviously it's a good example of a repurposing drug, and perhaps the biology will follow up. But again, this highlights one of the problems that we've got in rare skeletal diseases is a clinical endpoint for human trials. If you're looking at bone growth, it's fine in a mouse. So by 5 or 6 weeks of age, you know whether you've restored bone growth. But you can't wait for 10 years, I don't think, in a clinical trial. So we're really now beginning to think, how might we deal with that? And I think biomarkers is really the way forward. So this brings me back to this slide here. So I'll remind you, this was a meeting of several of these little people associations in the UK, really to come together and to try to identify what are the medical problems facing them and how might they best be addressed. We've really organised my colleague here in Newcastle, Michael Wright, and Melita Irvin, who's actually based down here in London, quite a few orthopaedic surgeons, some pain specialists, and then there's about 40 little people all came together and had a very active discussion on what was wrong and what needed fixing. The word research did not come up in the 2 days that we were there. Okay? So I had to give a talk at the end about some of our research approaches, and by then it had become clear to me that the term cure was not a good word to use in that meeting. The reason being, people with achondroplasia, there may be 4 generations, a family of 4 generations with achondroplasia, they don't have anything that needs curing. Being sure is not a problem, it's not an issue. So I had quite a good few conversations with some of these people. And basically the bottom line is that what they're wanting from research is very different from what we think we need to deliver. And that was quite an eye-opener for me, actually. So one lady said, if you can give me a drug that will stop my pain and stiffness, so she said, if you can give me a drug that will make my bones long, I'm not going to take it. You give me a drug that makes stops pain and stiffness, I'll take it. If that makes my bones grow long, I'm not going to take it. So it was really quite an eye-opener for me, and I had to start to rethink, really, what do we need to deliver for some of these patients? Now, I have to say that's probably specific to people with achondroplasia. And I'm trying to, there's one gentleman here, I think he's in about his early 40s, and he does a lot of exercise. He's never been to the doctor, whereas you see some of his friends are in wheelchairs and suffer quite badly from arthritis. But his major worry, so he does a lot of exercise, he does a lot of working out, and his major worry is he's got a teenage son, and he's encouraging his teenage son to do a lot of exercise. But he's worried, because he knows that his friends have cartilage degradation, he's worried by encouraging his son to do more exercise, he's going to exacerbate cartilage breakdown. So what he needs is some way, some indication of knowing when he's doing too much exercise and to stop. What he needs is a biomarker, basically, of cartilage breakdown. Which is really, so that's really focused our thinking. And we started to think that actually we do really need to put a lot more effort into looking for biomarkers, and fortunately we've just got a grant to do that. So we're going to actually spend the next 2 years trying to identify biomarkers for cartilage breakdown. Now, of course, this is where you get the link from rare disease to common disease. People have been looking for biomarkers for cartilage breakdown for 20 years, and I don't think there's a good one really for osteoarthritis as yet. But our thinking is, if you're looking for a biomarker for osteoarthritis, why not go to the really genetically tractable, severe phenotype, where you know that these people are going to have replacements by the time they're 15. So any cartilage breakdown is going to be occurring. It's going to be occurring very rapidly and easily detectable. So what we're going to do is we're going to collect urine samples from 50 patients with various achondroplasia, various chondrodysplasias, and 100 normal controls. And we're going to see is what cartilage breakdown product can we detect in the urine from these patients, and how applicable is that to end-stage osteoarthritis in a broader group of non-rare disease patients. So hopefully we can use rare diseases to inform on common diseases and actually come up with a biomarker that's really going to make a useful contribution to someone's quality of life. So with that, I will think that this is my final slide. So with regard to the broader field of skeletal dysplasias, we have made an awful lot of progress on genes and mutations. We are slowly beginning to tease apart disease mechanisms. That's not been easy to do because of the complexity of these diseases. But slowly, particularly with the ability to raise these large consortia with good EU funding, we have built up these cell and preclinical mouse models. We are doing repurposing, but I have to say, I think for some of our conditions, I think we're going to have to go straight for high throughput screening because we're looking at different compounds and we're just really not making any headway. So I think an unbiased, let's look at 10,000 compounds. We've got the readouts, let's look at 10,000 compounds and see if we can't identify some potential corrector molecules. And of course, biomarkers really underpins much of this work, and I think we need to put a lot more effort into thinking about biomarkers. Finally, just like to acknowledge the people in the lab who really do all of this work. Cassie's a lecturer up in Newcastle, Peter's postdoc, and students in the lab. But also had a very, very long-standing collaboration with Ray Boot Hanford down in my old university, Manchester, and much of the work that we've been doing on repurposing, we've been doing together, kind of comparing and contrasting different diseases. And Michael Wright, who's really the driving force, along with Melita in the UK, of really trying to bring, translational research to people with chondrodysplasia. And thank you for your attention.